Hi, this video I'd like to go over a few things with you in chapter 3, another chemistry chapter. But in this time we're looking at the chemistry of organic molecules. And so you can kind of think of it as biochemistry. And so there again, this is not really a chemistry class, but it is important to kind of go over a few things in the area of chemistry that really apply to living organisms. And so we'll go ahead and get started here in chapter 3. This outline just kind of shows you a brief overview of what we'll look at. First of all, really, what is organic chemistry? What are we talking about if a chemical is said to be organic, or what does that word actually mean? And then we're really going to go through kind of a step-by-step basis, talking about the 4 major categories of organic biomolecules or organic compounds that are found in living organisms. Carbohydrates, most of these you've probably heard about before, may already know a little bit about, such as carbohydrates, lipids, proteins, and also a group called nucleic acids. So the first section just kind of mentions a few vocab words for you. Organic molecules. When we hear that word organic, you know, it has other meanings in our day-to-day lives, because a lot of us realize that organic sometimes is referring to something that's natural or something that is grown in an organic way. It doesn't use extra fertilizers or extra chemicals or pesticides and things like that. And that's a perfectly valid definition. But in this case, we're talking, you see the word organic as it applies to chemistry or molecules in this case. It means that it has carbon and hydrogen as part of that compound. Now, it probably will have other atoms as well, other elements in that molecule as well. But by definition, it should have carbon and hydrogen as part of the molecule. And again, we're going to be talking about 4 main groups that are organic. A lot of times you see these called biomolecules, because they're the molecules that really make up a lot of the biomass for living organisms: carbohydrates, lipids, proteins, nucleic acids. So we'll go through that. Table 3.1, you don't have to necessarily memorize all of these functional groups. I want to outline a couple of these that you may see a little more often, a little more frequently. And if you go into chemistry, if you go into organic, and if you go into biochemistry, you would certainly need to know many of these functional groups. But a couple that you may see from time to time, for example, the hydroxyl group, the OH group. Anytime something is an alcohol, by definition, it's going to have that OH on the end. Sometimes we'll see this a little bit later on. We'll talk about the carboxyl group here. C carbon, it's double bonded to oxygen, and then it has an OH group also on that carbon. And you see that in your amino acid groups. Your proteins are going to have one end that will have that carboxylic acid group, carboxyl group there. Again, also the amino group, that's where you're going to find nitrogen as part of its molecule. So you have a nitrogen here and you have 2 hydrogens. And so again, a good place to locate that would be amino acids. I know we haven't talked about amino acids yet, but we will. And those are the building blocks to make proteins. And so those amino acids will have an amino group, and they'll have a carboxyl group associated with it. Sulfur groups, sulfhydryl group is listed there for you. And then the phosphate group, PO4, a lot of times we see this in nucleic acids. That includes your DNA and RNA, and then also ATP, which we'll get to later on. So what's so special about organic molecules that make them so very important for living organisms? Well, part of it is a few chemical properties associated with carbon itself, the carbon atom. It's a relatively small atom. It has 6 electrons. The atomic number is 6. So that means it's got 6 protons, 6 electrons. If it's neutral, then it will have 6 protons, 6 neutrons, and so it has a mass of 12. So that's very important. But also, it will form covalent bonds relatively easily. It can form up to 4 covalent bonds, and with all of these elements that are commonly found in living things. And we kind of mentioned that briefly in the last chapter: carbon, nitrogen, hydrogen, oxygen, phosphorus. and to some degree sulfur. It does create a covalent bond that is very stable, a carbon-carbon bond that is very stable. It can form chains, and so we'll see those chains, especially in things like fats or lipids. We'll see those forming there, called hydrocarbon chains. Not only can it form a carbon-carbon single bond that is shown here in this little section there, but it can also form a carbon-carbon double bond, and even a triple bond, and even can form a ring-type structure. And you'll see those again in the lipid category as we get down through the lecture. We'll see that as well. And it can also branch out, and so the chain doesn't always have to be straight. It can actually form a branch. And so there's a lot of different variations to the way that carbon can combine to itself. But again, one of the main things is that it is a very stable bond, and it does form those four bonds. Real quick, I backed up this table 3.1 because like if you look at the carboxyl group, each of these lines represents a covalent bond. And so you can kind of see the carbon atom there, and it has one covalent bond to this oxygen. It's got a double bond. So if you're talking about a double bond in organic chemistry, it'll be represented by two lines. And so that's two covalent bonds there. And so it can actually bond twice there. And then it can combine or bond to something else. In this case, the R represents other atoms that it could potentially form a bond with. And so then it could form a bond there. So you could look at this and say, okay, this carbon has four bonds. It's got two covalent bonds to this oxygen. It's got one to the hydroxyl group here. It's got one to something else, some other atom there, perhaps. All right, so there's your technical book definition of the functional group. Kind of looked at the table already. Clusters of specific atoms bonded to the carbon skeleton. But that really helps define what types of characteristics and properties that group is going to look like. Sometimes you see carbon and organic molecules having isomers. And so what that means is they have different arrangements in that molecule. They'll have the same exact formula. So in other words, these two are two different molecules. They have different characteristics. But if you went through and counted up the number of carbons and the number of hydrogens and the number of oxygens, this compound here would have the exact same number. So it has the same molecular formula, but because it's arranged differently, that actually gives it different chemical properties. So it's another important thing that carbon can do. Now, again, we're going to be looking at our focus really is carbohydrates, lipids, proteins, nucleic acids. One thing to realize is that these compounds mainly consist, especially like the carbohydrates and proteins and even the nucleic acids, are made up of individual units that all link together, all build together. So, and I'll show you examples of this as we go through, but that individual unit is called a monomer. Mono means one. And so the simplest part of that structure is known as a monomer. If you're talking about proteins, I'll give you an example here. If you're talking about proteins, the simplest repeating unit of a protein is known as an amino acid. That would be the monomer for a protein. Once you start putting those monomers together, you build up into form a polymer. Poly means many. And so you have many of those amino acids linked together, not just connected, but they are bonded together. There's a special bond that's going to form, but you bond those together and that forms a polymer of amino acids, and we call that a protein. Carbohydrates do the same thing, and we'll see those examples as we go through. Lipids are a little different. They don't exactly have that same structure because they have different subunits that make up lipids, and I'll show you that as we get to that section. Little table here, 3.2, helps you kind of keep all that straight. They give you the monomers there, the monosaccharide for the carbohydrates. That's the basic subunit, and you put monosaccharides together and you form a polysaccharide. Lipids, again, they still have subunits. It's glycerol and fatty acids. We'll see that as we get to that section, but it really doesn't form a polymer. It doesn't really form a chain like the other three do. Proteins, building block, the basic subunit is an amino acid. It forms chains called polypeptides or proteins. Nucleic acids, the basic building block is a nucleotide. And as we get to that, you'll see that that forms chains of polymers known as DNA or RNA. Good table to study, kind of keep things straight for the biomolecules. Now, also, we have to realize that your cells in your body is always, living things are always building things up and breaking things down. And so you have different types of chemical reactions, synthesis reaction, degradation reactions. And so the first vocab word here, a dehydration reaction, you kind of see the definition there. It is a type of chemical reaction where you're basically building something. This is a synthesis type reaction. But what do you think of when you think of dehydration? Think of losing water. Well, that's what happens in this chemical reaction. You combine things together. You're building something, but you produce water in the process. It produces H2O in the process. But it's a reaction that's actually making these polymers. You take those basic subunits, you put two amino acids together in a dehydration reaction, and you build up and you start the process to make a polymer or polypeptide or protein, as it is called. Hydrolysis is kind of the opposite. By the way, the ending of the word here, lysis, that's a good vocab suffix, I guess, the ending word there to make a note of. Lysis means to break apart, to split. And so hydrolysis means you're breaking something apart, and you're basically using water to break that molecule apart. And so this is how you degrade something. You're taking a larger polymer, breaking it down into smaller units. Good example is when living organisms, including us, when we digest starch. Maybe you have some type of food that has starch in it. You need to digest that, break it down into smaller sugar units, glucose units that you can digest easier. And so you would go through a hydrolysis type reaction to break it down into smaller and smaller subunits. Enzymes, we've got a whole nother chapter dealing with enzymes. We'll just say a couple of things here. They are required for cells. It does help to speed up a type of chemical reaction. So in other words, an enzyme, a lot of times you'll see the phrase, it's a biological catalyst. In chemistry, a catalyst is a molecule that helps to kind of speed up that reaction. Living organisms, we need enzymes to help speed up certain types of reactions that would not normally take place at our body temperature. And so we need to kind of speed up some reactions. And so enzymes have special characteristics in that, one, they're not consumed in their reaction. They're not really part of that chemical formula. They play an important role, but they're not really part of the chemical reaction itself. They don't get changed. They don't get affected. In other words, in theory, you could reuse an enzyme over and over again. And essentially, they are chemical catalysts or biological catalysts that help to speed up reaction. We'll talk about enzymes, give more examples a little bit later. All right, let's look at carbohydrates. This is something that you may be familiar with. You may know some examples. Some of the main functions of carbohydrates would be to provide some energy for the cell. And it's usually a very quick energy. Now, you can get energy, and you do get energy from lipids, and you can get some energy from proteins, but your main energy source really normally is some type of carbohydrate. In certain, they do form, in certain cells, they do form a structural role, is more so than others. In certain organisms, such as plants, you find complex carbohydrates in their cell wall, or in bacteria in their cell wall. And so depending on what organism you're looking at, they play a structural role as well. Generally, you're going to find carbon, hydrogen, and oxygen in carbohydrates. By the way, that's kind of what the word is telling you up there. If you've never noticed that, carbo refers to carbon, hydrate refers to water, and so of course that's H2O. So carbon, hydrogen, and oxygen are the elements or the atoms there for carbohydrate, and it's always in a ratio of 1 to 2 to 1. And these are the main Building blocks here, monosaccharide, that's the simplest monomer. If you put two monosaccharides together, by the way, mono means one, saccharide refers to sugar, and so one sugar unit. If you put two of those together, you form a disaccharide. Di means two sugar units or two monomers attached together, you form a disaccharide. After that, you just have really a polysaccharide, which means many sugar units put together. And a lot of that's here on this slide. Monosaccharide, single sugar unit. Good example of a monosaccharide is glucose. Okay, a lot of times we think of glucose as blood sugar. Our body is trying to break down carbohydrates to get it down simpler to glucose, and that's the energy that your body is going to use to convert that to, or your cells will use to convert it to ATP energy. That's another chapter, we'll get into that later. But glucose is a monosaccharide. The sugar that is naturally found in fruit is called fructose. It's also a monosaccharide. In DNA, you have monosaccharides called ribose and deoxyribose. It's also giving you some general terms. So glucose happens to have 6 carbon atoms, and so it's referred to as a hexose. The prefix hex means 6. Your ribose and deoxyribose that are in your nucleic acids forms, those happen to have 5 carbons, and so that would be a pentose type sugar. By the way, if you haven't noticed already, all of these sugars end in OSE, and that's a good indication that you're dealing with some type of sugar unit, the OSE ending. Disaccharide is what it sounds like. You take two monosaccharides, you join them chemically together, and so that would be an example of a dehydration type reaction, and you form a new sugar unit, for example, lactose. Sometimes you may be familiar with lactose. You may have heard of individuals that are lactose intolerant or have problems digesting lactose, milk sugar. That's because you have to have an enzyme there to break down the bonds holding these two sugars together. Galactose and glucose are the sugar units that have to combine to form lactose. Well, your body has to be able to digest that and break those back down. And so some individuals have a little problem digesting and breaking down the lactose. What we normally call table sugar, that's sucrose, and it's basically a disaccharide of glucose and fructose. Polysaccharides, poly means many, and so once you put together many of these monosaccharides, you form sometimes we call these complex carbohydrates. Complex carbohydrates, you've got lots of these sugar units put together. Good examples here. 3 good examples are listed there. We'll talk about the other 2 in a minute, but the 3 good examples here: starch, glycogen, cellulose. Make sure you have an idea of what these are. Starch is how plants store extra energy. They take glucose, they chemically combine them together, and they form starch. We kind of do the same thing. Animals do the same thing. Humans do the same thing, except we change the molecule a little bit. Instead of forming starch, we form something called glycogen. It's a polysaccharide. It's a very energy-rich compound but it's also an easy way to store extra glucose. Cellulose, this is more of a structural component, and it's found in plant cell walls. Now, we can't directly digest cellulose, but what we use it for is something called fiber, and that helps in our digestive processes. Chitin, this is another polysaccharide that you find in cell walls of fungi and in the outer exoskeleton in things like insects. Peptidoglycans, these are complex carbohydrates or polysaccharides that are found in the cell walls of the bacteria. Lots of examples of polysaccharides. Now, let's look at lipids. Again, most everybody has probably heard of lipids. You may know a few examples. We'll look at some examples. Not all lipids are the same. They do have a variety of their structure and their function a little bit. A couple of things about them. They're relatively large biomolecules. They're usually they are nonpolar by definition, and so that means they don't dissolve in water. Okay, so they're all going to have these characteristics. But look at the functions. They have a little variety of function here. They do store energy. Some of them do. Long-term energy. So again, let's say you have glucose as an energy source, but you have extra glucose. You can store that as some glycogen. Well, what happens if you have plenty of glycogen? Now, what do? How do you store that extra energy? You can convert that into lipids and store that in your tissues as fats. They do form structural components in cell walls and in cell membranes. And we've got another chapter dealing with the cells, and so we'll talk about some of the main lipids that you find in the cells. They help to retain heat for organisms, living organisms. They can actually help in cell communication and regulation because some lipids are hormones, and that helps to relay information and communicate different signals to different cells, and even provide a little bit of protection for the cells as well. They come in different varieties. Most of us are familiar with at least the first two, fats and oils. Those are both lipids. Phospholipids, you may not have seen that. We'll talk about that more in another chapter because that's what you find in cell membranes, the phospholipid bilayer. They also include a group of compounds known as steroids. We'll talk about those a little bit, and also waxes. Table 3.3 kind of outlines kind of what I just talked about a little bit. It's a nice table. It's a good table to study just because it kind of puts everything there for you. It gives you examples to look at, gives you the types, the main types of lipids here, gives you the kind of the main function for each of those types, and some common areas where you may see that. Let's look at some of these examples then. So one type of lipid is a triglyceride. If you go to the doctor and you get your blood work done, blood panel done, they're probably going to tell you, hey, this is your triglyceride level. These are the fats that you have kind of in the bloodstream, are triglycerides. And they do provide energy storage. You have specialized cells called adipocytes that are designed to store these energy reserves. And they do provide us a little bit of protection, like in other words, they can cushion our organs, give us a little protection there, and insulation, keep our internal body temperature hopefully close to that 98.6 degree area. The main structure is a glycerol molecule combined with 3 fatty acid molecules. Now, fatty acids, which I'll show you a picture of here, and I'll kind of go back to the notes. Fatty acids are basically long chains, long hydrocarbon chains. And as the notes up here say, that fatty acids can either be unsaturated or saturated. You may have heard of these words before. If you've looked on a nutrition label somewhere, maybe you've taken some nutrition classes, or you looked on a package and you saw the little label there. It may say, it has unsaturated fatty acid or saturated fats or whatever. Here's what this is talking about. Unsaturated has one or more double bonds between the carbons. Remember we said that the carbons can form single bonds, they can form double bonds, they can form triple bonds. Well, if it has at least one or more double bonds there, that's unsaturated fatty acid. So it's really a organic chemistry term. So if we look at the second picture or the second diagram here, you see the fatty acid hydrocarbon chain. We notice that there's some places here that have a double bond between the carbons. It's kind of small here. Take a look at figure 3.9 when you get a chance. You'll see the double bonds showing up there. The saturation here means it's saturated with whether or not that molecule is saturated with hydrogen. So in other words, if you look at the unsaturated fatty acid, it's not saturated with hydrogen. There's some spaces here because you can't have more than 4 bonds to that carbon. And you can count them up. I've got 1, 2, and then the double bond counts as 2. So I've got 4 bonds there. I can't add any more hydrogens to that carbon. So this is unsaturated. That's different than this molecule at the top. This fatty acid is saturated with hydrogen. I've only got single bonds all the way through. In other words, I can't add here any more hydrogen because it's all completely saturated. What that means for us is that unsaturated fatty acids usually are liquid at room temperature based on their chemical structure. They're normally liquids, so they're normal. We would normally call those oils. Vegetable oil, corn oil, peanut oil, these are all liquid at room temperature. Saturated fatty acids tend to be solid at room temperature. Things like butter, real butter, lard, animal fat, those things are saturated. And so from a dietary nutrition standpoint, the more unsaturated it is, the better it is for you. So sometimes you'll see the word polyunsaturated, which means many of those double bonds associated with it. And those oils are even better for you. And so things like olive oil, those are polyunsaturated, and those are actually healthy for you. Phospholipids, another type of lipid. You find these in membranes. We'll talk more about this when we get to the chapter dealing with cell membranes and things. But it's also glycerol molecule. It's got two fatty acid groups here. So it's similar to the triglyceride compound there, but its main function is to help form the membranes of cells. And so these are required to form the structural component of cells. And this is what that phospholipid would kind of look like. You have long fatty acid tails here. This is a nonpolar portion of it. These are called hydrophobic. It has a polar area here at the molecule where it has phosphate there. That kind of gives it different characteristics. This is called hydrophilic. The hydrophilic end is water soluble, and then the hydrophobic tail there is nonpolar, and so that's lipid soluble there. And so for a cellular membrane, and again, we'll see this a little bit later, just kind of getting ahead, but for the cell membrane, these lipid tails are here it's a double membrane system. It's two layers of these phospholipids. So outside of the cell here with the here, and then the polar water-soluble area here, and then you've got the lipid portion in the middle of this lipid bilayer, two layers of lipids there. Steroids, that's another group of lipids. These form ring type structures. I mentioned that at the very beginning, but carbon can form ring type structures. This includes things like cholesterol. Now, cholesterol gets a bad name a lot of times, and it can be certainly bad, but it is required for your cell membranes. It is very important. It does have some important roles. The problem is many times, if we eat meats and animal products, they have a lot of cholesterol there. And so our bodies usually make enough cholesterol, but a lot of times we eat a little bit too much, or we can't process the amount that we do have. So it does, and it can cause some problems. Testosterone and estrogen, those are reproductive hormones. And so those are steroid-based hormones, and they form these carbon ring structures. Cholesterol is also used not only in cell membranes, but it's also used to help form other steroids, including the testosterone, estrogen, progesterone hormones. And there again, too much can have bad effects because it can lead to cardiovascular problems. Waxes, you don't see a lot about this, but technically this is a lipid as well. It is nonpolar. They are they are waterproof. They don't combine with water. They're lipid soluble. They are lipids. They are solid at room temperature. They're basically a protective type function. Humans produce a little bit of earwax, and that's basically what's called cerumen. It's a type of wax that we naturally produce as a protective feature for our ears. Plants have a similar type of a wax that kind of creates a waterproof coating on the outside. All right, let's look at a little bit of proteins. Proteins are, we've already mentioned, are a type of, are formed from a polymer. The subunit is an amino acid. That's the smallest subunit of a protein, so make sure you make a note of that. Amino acids, they combine together to form eventually polypeptides or proteins, and they have a specialized bond called a peptide bond. So anytime you see peptide, that's referring to that special bond that makes proteins. It's a covalent bond, so it's a very strong carbon-carbon bond, and that will take amino acids, combine them together, and that starts your building blocks to form a polymer of proteins. There's many different types of proteins, and we'll talk about some of the examples and what some of those examples do. Sometimes you'll see just the word peptide by itself. In other words, if you just take two amino acids, sometimes you'll call those a dipeptide. If you put two amino acids together, link those together with a peptide bond, you would have a dipeptide. If you have three of those linked together, that would be a tripeptide. Really, after that, it's just referred to as a polypeptide, or once it's folded, once it has a structural 3D conformation, we're going to talk about that, then it's referred to really as a protein. Look at all these different functions. We won't go through all of these really right now, but proteins have a lot of different functions. Most of your enzymes in a living organism are proteins, so that helps with metabolism. You may have heard of keratin that you have in your fingernails and in your hair, the collagen that you have in your skin. These are proteins, so that helps support cells and support tissues. There are transport proteins. We'll talk more about those in another chapter, but cells have to be able to transport things in and out of the cell. Sometimes you have to have proteins to create a channel through that. If you've heard of hemoglobin before, this is a protein that's in your blood. In fact, it's in your red blood cells, and it helps to transport gases, carbon dioxide and oxygen, through the bloodstream. Your antibodies are proteins. That helps your immune system, so that's part of defense mechanism. Regulation, your hormones, many times are proteins, not all of them. Some of them are steroid-based, some of them are lipids, like we mentioned before, like the reproductive hormones, but many of your hormones are protein structure. Some organisms have a specialized type of protein called a microtubule that kind of helps that cell to move around. It almost like forms like a tail for that cell to actually move around. We'll see examples of that as we go through this semester too. Let's look at the basic building blocks here. The monomers, amino acids. Remember the functional groups I mentioned at the very beginning. You see those are listed here for you, maybe in a little different structure here, but you have an acid group. This is carboxylic acid. C, they don't show you all the bonds here, but C is double bonded to an oxygen there, and then into the OH group. And then you have the amino group is here. So all of the amino acids, there's basically 20 main amino acids that you find commonly in living organisms. And they would all have this basic type of structure. They all have a hydrogen here, you have a carbon, you've got the acid group, you've got the amino group. The R means that could vary. That could vary depending on which amino acid you look at. And figure 3.14 has many of the common amino acids. You don't have to memorize this. You can kind of take a look at it. But if you do take a biochemistry class, or if you go into chemistry, you'll probably have to study this in more detail. Shapes of proteins. Not all proteins have the same shape, but the shape is very important, mainly because of this first statement. Proteins cannot function properly unless they have the correct shape. They have to have the correct, what's called the configuration or conformation. If they don't have the right shape, they don't function correctly. When they lose their shape, that is a process called denaturation. They become denatured. There's certain disorders and diseases we may see in the chapter or in some of the chapters we look at that are based on a misfolding of a particular protein. If it doesn't have the correct shape, or something causes it to lose its shape, then it will not carry on its function. Proteins seem to be very susceptible to things like pH, acid and base concentration. Some proteins work well at a lower pH. You have enzymes in your stomach that work at a very low pH so that it can digest certain things. Most proteins, most enzymes seem to work better at a more neutral pH. Temperature is another thing. Most proteins don't work well in a higher type temperature. And so if you have the temperature too high, that can disrupt the protein. We'll have some labs that we experiment with enzymes and changing temperature, changing pH, and see what the effects are. Forming levels of structure that you're going to find for proteins, and they're listed there for you: the primary structure, secondary, tertiary, and quaternary. By the way, these words mean structure 1, first level, second level, third level, fourth level structure. And I know this is a lot of information here. Let's see, I think I have a couple of diagrams we can look at. Let's kind of look at the figure here first, 3.16. The primary structure, if you could just imagine putting all of the amino acids together. Primary structure is basically amino acids all linked together in a pretty much straight type chain. There's some little curves there, but pretty much a straight type chain. That's your primary structure of protein. The secondary structure, you can imagine this chain now kind of curling up a little bit. And a lot of times that's because there are hydrogen bonds that kind of reach out and kind of attract the neighboring section of that protein structure. And it kind of creates a coil effect or a helical effect. Sometimes instead of forming a helix, this is called an alpha helix, instead of forming a helix, it almost forms like a bent sheet, what's called a pleated sheet. And you kind of put pleats. If you fold paper up a couple of times, you form some pleats in it. You have a different type of a structure, but both of these are examples of secondary structures. Those secondary structures are because of the hydrogen bond attractions between different sections of the primary structure. Primary structure, you got a straight line sequence. Secondary structure, you've got a little attraction from one section to the next from the hydrogen bonds. Tertiary, if you notice the pattern, we're getting a little bit more folded, a little bit more complex. Tertiary means you kind of have a combination of the helical structure and the beta pleated sheets, and you got a little bit more interaction. The word I'm trying to look for, a little bit more interaction between the different sections of the secondary structure becomes more interactive. Tertiary structure is the overall three-dimensional shape now. Quaternary structure is when you have multiple subunits. That's the most complicated, not complicated, the most complex, the most developed form of the protein. And in this example here, it's the quaternary structure. There's actually 4 subunits. It's like taking the tertiary structure, but now I've got multiple tertiary structures all in one unit. That's your more complex type of a folded type protein. But now, like I said, if you can imagine, if this protein was exposed to extreme temperatures or acid conditions or the wrong pH, then that would disrupt its three-dimensional structure, and it may start unraveling or coming apart, and then it starts to lose its function. All right, last couple things we'll look at here. We need to talk a little bit about the nucleic acids. And so your 2 main nucleic acids we need to mention are DNA and RNA. There's whole other chapters. We've got a unit on genetics that we'll get to later, but just a couple of things about nucleic acids. One is your basic subunit is known as a nucleotide. Nucleotide, we'll see that in just a minute. DNA, of course, this is deoxyribonucleic acid. This is your genetic information that is stored inside of the cell, inside of the nucleus, and that gets passed on, that carries on that genetic information to the offspring. It also carries the information on how to form certain amino acids and how to form proteins, and that gives us all of our characteristics. RNA, ribonucleic acid, that does other things, but it helps regulate processes known as protein synthesis, making proteins, and gene expression, which genes are turned on and which genes are turned off. So again, we'll have more of that information as we go through. Right now, let's look at the structure of a nucleotide. One thing you need to know is the three main parts: a phosphate unit, and that's PO4, that was one of the functional groups at the very beginning. A sugar unit, we talk about pentose, that's a 5-carbon monosaccharide, 5-carbon sugar, and then a nitrogen-containing base. All right, so each nucleotide will have these three components: phosphate, sugar, and a base. There are essentially 5 main bases that are located here, and so DNA has these are examples of the bases: adenine, guanine, cytosine, and thymine. Those are nitrogen bases. RNA has 3 of the same: adenine, guanine, and cytosine, but instead of having thymine, it has uracil as its 4th type of nitrogen base. Here's just a simplistic structure of all of this we just said. Remember the functional group, unit up here, the phosphate group. And so a nucleotide, by definition, is going to have a 5-carbon sugar. Sorry, that's the carbon base, the nitrogen-containing base, a 5-carbon pentose sugar, and then the phosphate. One way to remember this is, if you know the television station PBS, that's your abbreviation, PBS, phosphate, base, and sugar. Maybe you can, that'll help you out kind of remembering what has to be present in a nucleotide. Now, there's a lot of information here about the structure of DNA and RNA. The backbone of it is composed of alternating sugar phosphate molecules. Let me see if here's a kind of a picture of one here. So you've got the sugar and the phosphate, and it alternates. So this would be the backbone of that polymer. So each section here will be your nucleotide: the phosphate, sugar, and the base. Phosphate, sugar, and the base. So, but now we've got, in this example, we've got 4 nucleotides put together, and the backbone alternates between the phosphate, sugar, phosphate, sugar. RNA is a single-stranded molecule, whereas DNA is double-stranded. It's a double helix. There's 2 strands of DNA, 2 strands of the nucleotides in DNA that kind of wrap together. And we'll see that a little bit more as we get into the genetics section. Generally, the nucleotides pretty much go in any order. There are some basic rules that they have to follow as far as how they combine strand to strand. Like for DNA, adenine will make a hydrogen bond with thymine. Cytosine will make a hydrogen bond with guanine. There again, this is getting really a little ahead of the game because we just want to kind of introduce these as a nucleic acid, as the nucleotides, understand a little bit about these, but we're going to get into more of this detail as we go through the structure, as we go through the unit and get into the genetics unit. Table 3.4, that's actually a nice table because it kind of gives you a quick look at DNA and RNA and compares what's there. So each one has a 5-carbon sugar. DNA has deoxyribose, RNA has ribose. Each of them, because their nucleotides are going to have bases or a base, and it lists the 4 bases for DNA, lists the 4 bases for RNA. Again, the main difference is RNA has uracil and DNA has thymine. It tells how they kind of, the strands match up. For DNA, it's going to be double-stranded. For RNA, is single-stranded. Most of the time, it's single-stranded. For the DNA, because it does form a double strand, in other words, two strands form together, and then there's a kind of a twisting of that, again, because of the hydrogen bonding. It twists together, and that forms a helical or coil type shape. RNA is pretty much linear. Yeah, there's your kind of the diagram in figure 3.2 of the DNA. Maybe you've heard it called a double helix before, right? It's double-stranded, 2 strands of nucleotides, and it forms a coil that goes around. The last thing here we need to look at is ATP, and we're going to spend some time talking about this molecule a little later on as well. This is This is made out of nucleotides, so it has the same type of composition. It has adenine in it, one of the bases. It's got a 5-carbon sugar, it has ribose, and it has a phosphate group, but actually ATP, the word up here, has adenosine triphosphate, so that means it has 3 of these phosphate groups attached. So it's made out of basic nucleotide fashion, but it doesn't make the polymer like DNA and RNA do. But it's very important for all life forms because this is the energy source our cells need to complete any kind of cell reaction or the work inside of the cells. A lot of times you see ATP called the energy currency of the cell. This is your energy unit that your cells have to go through any kind of chemical reaction inside of the cell. And so we're going to talk later on about energy reactions, how cells get this ATP and kind of what they do with it. Sometimes you'll see this word ADP. This is adenosine diphosphate. There's only 2 phosphate groups attached here. And one thing that we'll see is that the energy associated with these molecules comes from those phosphate bonds. So if you have the 3 phosphates attached, that's your higher energy molecule. There's only 2 of those attached, then it's less energy. So we'll see that a little bit more as we go through and we get a little closer to the unit on energy. And there's kind of a diagram figure for ATP. All right, guys, thanks. I hope this was helpful for you. I know it's a very quick overview of the chapter, but maybe you were able to learn something from the chapter there. Please look at the vocabulary again. Find some way that helps you study vocabulary. There's a lot of terms. Use some of those tables because the tables really do a good job of kind of organizing everything into a small space. Use the tables to help study. Make some flashcards. Write out carbohydrates and lipids, and see if you can come up with the basic function. Maybe some examples kind of help you out where they're located, where you find those, but really focus in on some of that vocabulary, and that'll help you out as we get closer to exam time and as we get closer to some of the other chapters as we move forward. Thanks, guys.